Thank you for joining the Naked Immunology webinar series, and apologies for the small delay. We had some technical difficulties, but now everything is in place. Um, I'm going to short the introduction, so it's my great pleasure to welcome Professor Soman Abraham from Duke University, who has been literally leading the field of mast cell biology for the last decades. And it's always inspiring, out-of-the-box thinking, and so we are really glad that you accepted to participate, Soman. And now the floor is yours, and I'm going to let you drive us through your nice presentation. Thank you, Soman.
All right. Thank you, Nico, for the opportunity to present my work. And again, I apologize for the delay here. What I was going to do was tell you a little bit about some of the work we've been doing pertaining to mast cells and food allergies and anaphylaxis. So a little bit about that. So food allergies affect about 4 to 10% of the global population, and it's increasing in both children and adults. And what's interesting is there seems to be a link with bacterial infections. It seems bacterial infections either precede or the food allergic reaction, or there's some changes in the endogenous microflora that might contribute to the anaphylactic reaction. Now, unlike airway or skin allergies, food allergies often have rapid onset and have life-threatening anaphylaxis. And so that way they're quite different from allergies that impact, as I said, areas such as the airway and the skin. And what I was going to do today was actually describe 3 observations that we've made over the last several years that provide some clues as to why food allergies are life-threatening and why there's a sudden onset. And they all seem to have something to do with the mast cell. So why are food allergies uniquely associated with life-threatening anaphylaxis? So there's been a long-standing notion that following food allergies, following ingestion of food allergens, the allergens quickly get into the blood. And then once they are in the blood, they are then able to activate the mast cells throughout the body because of the close association of the mast cells with the blood vessels. I was wondering if you can see my pointer. Do you, by chance, can you see it?
No, we do not.
Can you see my pointer? I guess I can't hear you. Okay. Okay. Yeah. All right. So these are it's a picture of mast cells found around the blood vessel, and when allergens get into the vasculature, they're able to activate mast cells throughout the body. And when that happens, the mast cell products then induce vascular dilation and leakage. So the result is there's a huge drop in blood pressure and also a drop in body temperature. And that's often associated with anaphylaxis. Okay. So this is a picture of a mast cell before degranulation, and this is a picture of the mast cell after degranulation. And what is most striking is the large number of granules that are released. And this is just a diagram showing what happens when IgE and allergens cause mast cell degranulation. You have IgE bound on the mast cells, and when allergens bind to them and cross-link these receptors, there's this spontaneous and rapid release of granules. So a lot of these granules are released, and the key difference between this event and what happens, say, in a macrophage or other innate immune cells is that the release of mediators is slow and over a long period of time. Here is a rapid release, which occurs within a few seconds to minutes. And often the products contained in these granules are serine proteases, such as chymase, and histamine, TNF, and so on, various cytokines, most of which are pro-inflammatory. And here's a picture of a single mast cell granule. And we have shown that these granules don't break up once they're released. They're actually highly stable. And so they can remain as slow release factors. They can release the contents over a given distance. And these granules also can traffic great distances to go further away and impacts its effect. So what I'm going to do today is tell you about studies where we use this mouse model of systemic anaphylaxis. So basically what we do is we infuse mice with IgE antibodies against a particular allergen. And so the antibodies bind the the antibodies bind the mast cells, and then we add the antigen in systemically. So it's more or less just like what happens after you tested food allergens. The allergens get quickly into the blood, and then that activates mast cells throughout the length of throughout the vasculature, and often that is associated with a rapid drop in blood pressure. This is what you see when you have systemic anaphylaxis, and in a mast cell deficient mouse, you see this absence of this drop. And this is a picture of a mast cell sitting on the blood vessel with a lot of released granules. And this is a cross-section of a mouse where we've injected a dye into the blood system, and the green are the mast cells, the blue are the blood vessels, and the dye is kept inside the vasculature. But following degranulation, you can see the mast cell granules around the mast cells. So following addition of an allergen into the blood, the mast cells degranulate, and some of the dye you can see here beginning to come out of the vasculature, and it gets into the tissue. So this is the vascular leakage you see associated with anaphylaxis. So a key question we asked several years ago is, how are the perivascular mast cells systemically activated by blood-borne allergens? Okay, so remember, the mast cells are not in the blood, but they are outside the blood in the perivascular space. So how are they getting activated by the allergens? So what we did is we injected the allergens, which is in red color. So these are the vasculature with the allergens, and the blue are the mast cells. But we also noticed there's another cell sitting on the vasculature that seemed to pick up the allergens, not the mast cells. So the mast cells did not appear to take up the allergens. And let me see if I can get this. This video is not working, but anyway, you can see here that these are actually dendritic cells, and then you can actually see here the yellow, the dendritic cells taking up the dye. Okay, so the mast cells are the blue. They don't seem to take up the dye. So you're actually seeing here a yellow color, which indicates that the allergens are taken up by this cell and not by the mast cell. And what are these cells? So these are dendritic cells that actually sit on the vasculature, and they seem to stick dendrites into the vasculature and take up whatever is flowing through. So they serve as sample. They sample the blood. Demonstrate the importance of these dendritic cells in the anaphylactic reaction. We knock down the dendritic cells. We knock out the dendritic cells. So we got rid of the 301B positive dendritic cells, and we see a dramatic reduction in the anaphylactic response, indicating that, like mast cells, these dendritic cells were playing a crucial role in the development of anaphylaxis. Then the question is, how were these dendritic cells then transferring these blood-borne allergens to mast cells? And what we showed, this is actually a video tape here, where we are actually showing the green are the dendritic cells. And there's this dendritic cell which seemed to have sampled the allergen, and you can see that, and they are in vesicles. And in this particular image here, you see dendritic cell delivering allergens to the neighboring dendritic cells. So there is a transfer here that's occurring. Here you can see that here, and then it's finally taken up by this cell. In the videotape, it's a little bit better. And here is actually an dendritic cell, which you can see a little bit of yellow here. It's now beginning to pass it on this to the mast cell. So you can see the transfer that is occurring, and that's shown here as well. Okay. So you are seeing that transfer. So the vesicles are transferring the allergens which it has taken out of the blood and passing it on to neighboring cells, so including other dendritic cells as well as mast cells. And here, so what we did here was to see if we can block the vesicle formation in these dendritic cells. Can you prevent anaphylactic reaction? So what we did here was we depleted dendritic cells, the 301B dendritic cells from mice, and then we adoptively transferred wild type dendritic cells here, and unfortunately the video isn't working, but here we would have seen the vesicles being released here naturally. So these dendritic cells naturally release the granules. I'm sorry, not granules, they are vesicles which contain the allergen. Here is a knockout state where we knocked out VPS4, which is very important in the vesicle formation. So here we don't see any vesicles being released. Unfortunately, the video isn't working, so I can't show you that. You have to trust me. And then this is a quantification of the vesicle formation. There's a dramatic drop here. So when we infused these two dendritic cells types into the dendritic cell depleted mice, and then we introduced the allergen into the blood, we saw that in the knockdown, we saw a lot less vascular leakage, which is shown by the dye that is showing in the ears. We quantified that. So this is the control wild type, where we see very nice leakage and very little here. So what our data suggests that anaphylaxis is severe and systemic because blood-borne allergens are sampled by dermal dendritic cells throughout the body and quickly transferred to perivascular mast cells via allergen-loaded microvesicles. So here you have the dendritic cells. They are sampling the allergens, which go in the blood. They release vesicles which contain the allergens. They quickly go activate the mast cells. The mast cells then release mediators that cause vascular leakage as well as vascular dilation. And then in addition, some of these transfer the allergens to other dendritic cells, which can then traffic to the lymph node to present antigens. Okay, so this is the way that mast cells sample allergens that get into the blood. So another observation we made, so I'm going to the second story here, and that is the rapid onset and severity of anaphylaxis caused by blood-borne allergens could be related to a neuronal involvement, okay? So up till now, I was telling you that the mast cell mediators affect the blood vessels, inducing dilation and leakiness, giving rise to the anaphylaxis. But now I want to present data which suggests there's also a neuronal component here. And so here, what we see is, and the idea came from observing that, observing that mice that are going through anaphylaxis, it demonstrated a strange mouse behavior. They seem to lie prostrate. It's almost like a spread-eagled appearance, like here, if you're a human, this is how we behave. And this behavior was consistent with the behavior of mice that have been put at very high temperature, not very high, at warm temperatures. They seem to spread out their legs and hands. So this is the same behavior, which made us think that maybe there was a neuronal component in the anaphylactic response, right? So what I want to point out here is in our bodies, we do have a thermoregulatory neuronal circuit that senses temperature. So the peripheral neurons in the body senses the temperature of the outside and then sends message to this thermoregulatory system to either induce vascular constriction. In the case of when it's cold, it's vascular constriction, so you lose less heat, or as in here, when it's very hot, there is vascular dilation, which allows heat to be dissipated, and so the body cools down. So we do have that, and so do the mice that I will be describing. They have this a mechanism. And the other point I want to make is mast cells not only sit near the blood vessels, which are shown in pink, they're also sitting around nerves, which are shown in blue here. So potentially when mast cells degranulate, they can also impact neurons here. Okay, so I just wanted to bring that point up. So what we thought is to demonstrate the specific role of the nervous system in mediating anaphylaxis. And so we used a chemogenetic approach where we had these transgenic mice, which when you induce anaphylaxis will trigger neurons that are involved in anaphylaxis, and then there's a way to specifically label those neurons with a novel receptor, a unique receptor, and you can specifically label those neurons which get activated during anaphylaxis. And then later on, what you could do is you could then put a designer ligand for those neurons which are specifically activated, and then see if you can induce hypothermia or anaphylaxis without even giving IgE or activating mast cells. So the goal was to see if we can specifically activate the neurons that are involved in anaphylaxis and label them with this designer receptor, and then subsequently activate these designer activators alone and see if they can also give rise to the drop in temperature or anaphylaxis. And so that's what we did here. Oh, sorry. So here is, we had a whole bunch of other controls that I am not presenting, but we show here that this is what you get when you just activate those specific neurons which were labeled by that designer receptor. And when you activate that with that designer ligand, you can get anaphylaxis. And the control one had very little response. Okay, so by specifically activating the neurons that were labeled with the designer receptor, we could mimic the anaphylactic response, which then suggested to us that there was a neuronal component in the response. And what we know about the warm thermoregulatory neuronal activation is that there are peripheral nerves in the body which sense the temperature, sends message to the DRGs, the dorsal root ganglions, which then send signals to the spinal cord, all the way to the pons, and then to the hypothalamus, which is the thermoregulatory center in the brain. And then these then send signals to adjust the body temperature. So this is the thermoregulating circuit, and we want to ask the question, are these activated? Can we induce activation at these various sites? Okay, so so the first thing we did was to see if we could get labeling of the DRGs after we activate during anaphylaxis. So here is a picture of a cross-section of the DRE. There's a DRG, dorsal root ganglion, and these are the various neurons here. None of them seem labeled, but in the IgE overstimulated mice, the DRGs, about I think 14 or 15% appeared to be labeled, indicating that there was a signal. And these DRGs were in fact activated during anaphylaxis. In a mast cell deficient mouse, you don't see that. So we were able to show that this region got activated, this region on the thermoregulatory circuit got activated. And then the next one was the pons. And so here we again look and we see here labeling here in the anaphylactic mice, but not in mast cell deficient mice. So again, those the signals, neurons here were being activated in the pons, and then so typically what happens is after the hypothalamus is activated, they then send a signal to the brown adipose tissue, the back of the neck of the mouse, which then regulates the body heat. So if it's a warm signal that comes, it will suddenly shut down heat production in the back of the neck, and that can be easily measured. And if it's a cold signal, this back of the neck gets warm because that's because heat is generated there. So this is a picture where we are looking at mice. This is mice that are been exposed to saline. We see nice heating up of the brown adipose tissue. So this is normal. And then after IgE activation, so we see here that within the first few minutes, there's nothing here, but then subsequently we see that red disappearing, indicating suddenly the heat production has shut down. And if you actually monitor the temperature here, it goes down rapidly after IgE activation. So indicating that the brown adipose tissue is involved and it's gotten a message to stop producing heat. And that's why you see that rapid drop in temperature. Okay, so that's the signal that it got to reduce the temperature, and it involved the thermogenesis pathway. So what we are thinking is happening is during mast cell activation, following allergens which are in the blood, the allergens activate the mast cells, mast cell release mediators, which affect the vasculature, like I said, induce dilation and vascular leakage. But at the same time, it sends signals to the peripheral neurons, which then send signal all the way to the thermoregulatory system. Okay, so the next question is what neurons are actually activated here, and also what mast cell products activate the neurons? Okay, so typically for the thermoregulatory unit, there are several neurons which are involved in detecting the cold, and then there are other neurons which are more proficient in detecting warm temperatures. And specifically TRPV1 as well as TRPV4 are the primary neurons that are detecting warm temperatures. And so since we are interested in the warm response, we looked at these mice, and then we looked at TRPV4 and TRPV1 mice, and we see that there's very little difference when we use TRPV4 knockout mouse in the anaphylactic response between wild type and this, so indicating the TRPV4 neurons were not involved. However, the TRPV1, we saw a difference. It's not that much of a difference here, but again, remember, like I said earlier, the mast cell regulates drop in temperature and through activation on the neuron as well as the blood vessel and the vasculature. So what we are seeing is the specific effects on the neurons and not so much on the vasculature. And so the differences are not as high as you would expect. Okay. And here again, just showing the difference here. And then we also wanted to, since TRPV1 is found on all cell types, we wanted to make sure that it is the TRPV1 specifically found on the neurons that is involved in this temperature drop. And so what we used adenovirus that was which expressed the TRPV1 genes and associated with GFP, and then intravenously injected that into TRPV1 knockout mouse. And then what happens is once we inject them, these viruses will infect neurons and then induce those neurons to suddenly express TRPV1 And so after 4 weeks after injection, we're able to show that these TRPV1 repeated mice, if you will, are now able to respond to anaphylaxis. Okay, so the by giving that TRPV1 specific to the neurons, we're able to show it was the TRPV1 on the neurons that was specifically involved here. And the other thing I want to point out is that when mast cell products activate TRPV1 sensory neurons, I guess the question is, what mast cell products activate TRPV1 sensory neurons, right? So there must be something on the mast cell that specifically activates TRPV1. And one thing to also point out is when we activated the mast cells during systemic anaphylaxis, we see a lot of the granules falling not only on the blood vessel, but also on the neurons, right? So actually, when you use a scanning EM, you can see these are neurons with a lot of mast cell granules falling on them, indicating that perhaps that was the way mast cells were activating neurons. Okay, so to test that, we cultured these GCamp mouse DRGs. So these are specifically labeled DRGs, which when they get activated with the calcium flux, they light up. And so what we did is we cultured the DRGs and then placed mast cell granules on top of them. And then we looked and we see a good calcium response. And we subsequently followed this exposure with capsaicin, which is a very potent activator of TRPV1, and we show that the same neurons are involved in giving a very strong TRPV1 capsaicin response, indicating that these granules were in fact activating TRPV1. Okay, so the specific components on the mast cell that was activating were the granules by falling on the neurons that were activating the TRPV1. And so the granules contain a whole bunch of mediators, and we were curious to know which mediators were activating TRPV1. And we were specifically interested in the kinase. So what we did was we used kinase knockout mice, and we see a difference between the wild type here and kinase knockout. So the kinase knockout mice did not induce as big an anaphylactic response as the wild type, indicating kinase, which is specifically produced by mast cells, was involved. And so what we then did was to see how kinase was activating this TRPV1, because kinase does not necessarily bind or activate TRPV1. It has to do it through an indirect mechanism. And we looked at PAR1 receptors on these neurons. And here is the effect of kinase, again, on the same calcium flux assays. We see kinase here giving a nice response like the mast cell granules. And if you put a PAR1 antagonist here, we can block it. And then this is, again, a nice response that you see with just capsaicin. Okay, so we are showing that this response that we see here is actually through the efforts of kinase found on the mast cell granules, which act on PAR1 receptors, which in turn then signals TRPV1 on these neurons. And then this is the message that goes up, sending the message to the brain to then drop the temperature. And so you are able now to link the neuronal system to anaphylaxis, in addition to the previously described role of mast cell activation of the vasculature. And so I'll go now to the third part, which again is a slightly different story, but again, I will try and tie it with what I said earlier about the role of mast cells in trying to enhance the speed and severity of anaphylaxis. So before I do that, I just want to give you a little bit of an introduction to inflammasomes. So inflammasomes are found in lots of cells and is best studied in macrophages. So they are a collection of proteins that are found in the cytosol of the cell, which is important in the release of IL-1 beta. And it's a very important signal when innate immune cells are challenged from outside, either with pathogens, danger signals, and so on. And often what happens is when macrophages are, say, exposed to LPS, which is often the case like macrophages in the gut, where there's a whole bunch of microbes in the gut. The macrophages in the gut are exposed to the LPS, so they then start making pro-IL-1 beta in the cytosol, and they keep it there. And then when you get a second signal, such as an activation signal, a danger signal, they then form the structure called an inflammasome made up of ASC, NLRP3, and procaspase-1. This is a protease which is involved in the final clipping of procaspase to release IL-1 beta. Okay, so this complex called inflammasome is important in the release of IL-1 beta. I'm telling you this because I'm going to present some data to show some components of this inflammasome are also involved in IL-1 beta release during IgE-mediated degranulation. Again, so the two key points to remember is macrophages typically found in the gut and so on that have been exposed to endotoxin start making pro-IL-1 beta, and then subsequently these pro-IL-1 beta, upon activation with the danger signal, this pro-IL-1 beta is processed by a structure called a heteropolymeric structure called inflammasome. So one thing we noticed while undertaking our studies was if we pre-treated mast cells with LPS, just like I said before with macrophages, and then activated them with IgE, we saw something extraordinary. We saw a good level of IL-1 beta being released. And this is released within minutes as the degranulation is occurring. So actually, if you look in the mast cell supernatant, you see very good beta-hexosaminidase, which is a mast cell granule product. So the amounts of IL-1 are released around the same time as the granule contents are released. So for the first time, it looks like mast cells have the capacity to release IL-1 beta during degranulation. And that's because they were pre-treated with LPS. And what is interesting here is we see here, so the green is CD63, which is a marker for mast cell granule membrane. So you can see that in time 0, this is CD63. And then 5 minutes after activation with IgE, you see the granules now beginning to form to be released, and then they're all released here. And if we stain for IL-1 beta, we see that initially after LPS treatment, there's a lot of cytosolic IL-1, mostly in the periphery, but then following activation, you can see them all moving into the granules. You can see here the granules, these within a few minutes, 5 minutes of activation, you see IL-1 beta being processed, being trafficked into the granules, and then 10 minutes later they're all released, right? Because this is, you don't see much again in the periphery, whereas here we see a lot of IL-1 beta. So there is rapid release of IL-1 beta. And what is also interesting is this IL-1 beta, when you look at it, is actually when you treat these mast cells with LPS and then activate them with IgE antigen and look in the supernatant, we see both pro-IL-1 beta as well as the processed form, okay, in the supernatant, all right? And we then began to think if maybe it is the kinase in the granule, which is when the outside is actually processing this to form the complete IL-1 beta, the processed one, or active IL-1 beta. And here is some more additional Western blots showing what happens if you add chemostatin, which is a specific inhibitor of kinase. If you look here, you see both the processed and the unprocessed IL-1 beta in the supernatant. But if you add the chemostatin in the supernatant, you don't get any processing, indicating that everything remains in the pro-IL-1 beta form. All right. So it does look like the processing of pro-IL-1, unlike what you see in the macrophage, in the mast cell, is the full pro-IL-1 form that is actually released in the granule. But once outside, they're being processed. And this is more data here where we look at the supernatant and we collect the granules out. And then when we run a gel, you see that in the LPS treated one, when we collect the granules and spin them down and then look in the supernatant, okay, so this is granule remnant free supernatant, you see the processed one. Whereas if you look at the granules themselves, we see a lot of pro-IL-1, indicating that they are processed in the granule and then released into the supernatant. Okay, so there is actually a bolus of IL-1 released by degranulating mast cells, and which may significantly impact the pathological effects. And if you look in the literature, what is said about IL-1 beta is that they play a key role in breaking down tight junctions of epithelial cells and endothelial cells, which to my mind is very important because they can potentially, if mast cells are here, and this is the gut, and you have a lot of allergens here, mast cells releasing IL-1 beta here can induce leakage of the GI, of the epithelium here, allowing allergens to go in, and also the vasculature, it breaks down the tight junctions of endothelial cells as well, so allows the allergens to get into the circulation. And so this mast cell generated IL-1 beta potentially can induce rapid entry of the allergens into the blood. And that is why when you ingest peanut and so on, and if you're allergic to it, within minutes, you have a reaction. And we think that this mast cell products, specifically IL-1 beta, can have this effect. And then the other effect of IL-1 beta, which is well known in the literature, is they can lower the threshold of neuronal activation. So if you have IL-1 beta release, they can induce the neurons which are found here, they can trigger them to respond to even lower levels of activators. And so even small amounts of allergens, minute amounts, can trigger an activity by that. So, to test in vivo if increased exposure to LPS and the toxin could make the anaphylactic response more severe, what we did is we looked at the anaphylaxis of regular mice, where we see a good anaphylactic response. But if we pre-treat the mice with LPS, and then 4 hours later, activate them with IgE allergen, we see a much more severe form, and the mice actually die in this case. And if you add antibodies to IL-1 beta, if you pre-treat these mice with antibodies to IL-1 beta, you can actually protect some of them from death. Okay, so the severity of the immune or the inflammatory response is critically determined by IL-1 beta. And that we think also is a contributor. And then finally, I will just tell a little bit about some additional observations we made, and which is that this trafficking of IL-1 beta to the outside is actually mediated by two components of the inflammasome system, NLRP3 and ASC, but not the third component, caspase-1, in the mast cell. And so we made this observation where if you knock out NLRP3 or ASC, either one of them, you can dramatically reduce mast cell degranulation. So this was a very striking finding because it suggested that these cell proteins which are normally involved in releasing IL-1 beta are actually involved in the mast cell degranulation response to IgE. And whether or not there's IL-1 beta or not, these components play a crucial role, which has never been shown before, in the degranulation process. So if you use knockout mice of NLRP3 or ASK, you don't get much anaphylaxis, and this is the wild type. And if you use mast cell deficient mice that we had infused with wild type bone marrow mast cells or with ASK minus or NLRP3 minus mast cells, you get no response here. Whereas if you use wild type mast cells, you get a nice degranulation and anaphylactic response, indicating that these inflammasome components actually have a role in mast cell degranulation, but it does not involve caspase-1, which is the other component of the inflammasome. And the reason we believe is that because the mast cell granules contain its own protease. Caspase-1 is a protease that cleaves pro-IL-1, but in this case, the mast cell has kinase in the granules, and so this processing can occur. And so this is what we see in the regular macrophages. IL-1 beta secretion involves this three components, ASK, NLRP3, and procaspase-1, whereas in the mast cell, IL-1 beta release involves the movement of IL-1 beta, pro-IL-1 beta into the granules, and this whole movement of the granules is mediated by NLRP and ASK, which together bind CD63 and then traffics this on the microtubules all the way out. So this is a novel finding demonstrating mast cell degranulation is mediated by some of the inflammasome components. So I will end here by summarizing our findings, which is perivascular dendritic cells present allergens to mast cells, and that is the reason why we have rapid amplification of systemic anaphylaxis. So when allergens get in the blood, the perivascular dendritic cells quickly sample them, pass them on to mast cells, and this happens throughout the body, and that's why you get this amplified effect. The thermoregulatory neural circuits triggered by mast cell degranulation further accelerate anaphylaxis. So there's a neuronal component, not just the vasculature, that is involved. And then the other point I want to point out is endotoxin primed mast cells release IL-1 beta during IgE-mediated degranulation, which can enhance the leakage of the endothelium as well as the epithelium of the gut, causing greater entry of the allergens, and so you get a more intense reaction. And then mast cell granule remnants are they serve as extracellular hubs for the processing of pro-IL-1. And when this happens outside the body, you tend to have a very rapid release of IL-1 beta, and it's highly localized, and that can potentially amplify inflammation. Because when you have a macrophaging release IL-1 beta, it takes hours for a significant amount of IL-1 beta to be released. Whereas with the mast cells and the capacity to release to degranulate, within minutes you get a large bolus of IL-1 beta, which can significantly impact the pathology. And finally, therapeutic targeting of NLRP3 has been shown to prevent mast cell degranulation. And similarly, IL-1 beta antagonists or receptor antagonists have also been shown to modify the anaphylaxis to bring it to a less severe form. So these are some other observations. So I will end by acknowledging the key people who did the study. So the stories with the neuronal involvement involved Evangeline Bao was a graduate student. The dendritic cell sampling story was by Hae-Woong Choi. He's now a faculty in Korea University, and Evangeline works in industry at Roche in China. And these are postdocs who did the IL-1 beta story, Andrea and Pradeep. Thank you so much for your patience. Thank you.
Thank you very much, Soman, for the very comprehensive set of data and for this tour de force to present all those data in a short amount of time. I think that's we have many questions for you actually from the audience. So I'm going to start by addressing
Nico,
them
we
one
can
by.
hear you.
Yeah, can you hear me?
And my mic is, I wouldn't be able to hear Nico's because I can't.
1 second,
Could
sorry.
You may have to repeat the question.
Sure.
Nico, can you repeat the question?
Yeah, can you hear me now? Can you hear me? Hello?
Sorry, there.
Yeah, now it's working.
Yes, I can hear you now.
Okay. No, I would say thank you so much for this great presentation. And so we have many questions and not a lot of time, so I'm going to just jump into it from the audience. We have a first question from Alina Gravelov saying, which mast cell efficient mice did you use for the GRG neuron project? Did you use kit-sash mice or Cree mouse lines?
So we use different mast cell deficient mice. We used MCPT5 knockout for certain studies. And then we use also mast cell deficient mice, the sash that we repeated with wild type or knockout mice. So we used various ones, but it's actually in the paper. So you can look at that, yes.
Excellent. Another question from Philipp Starkel from Vienna. It says, hi, Soman, many thanks for the great talk. So what is and I think this is a fascinating question as well. What is the physiological function of the anaphylactic shock drop in body temperature? So why would you need to immobilize yourself, actually, if you think that you are in the wild and that it's a survival actions?
Okay. All right. So this is my opinion. So the neuronal network and so on, their main goal is to, when you get injury on the skin or some sort of thing, they will send itch response, they will send pain response to the brain, and that serves to enable the host to respond in some way. Okay. But the problem is, if the signal now, the poison, if you wish, the allergen now, if it's no longer on the skin and so on, but actually gets into your blood, then what the body is trying to do is it's now thinking we have poison in the blood, right? So you don't want it to go to the critical parts of the body, such as the brain and the major organs. So what does it do? It induces vascular leakage so that the poison is leaking out. So enough concentration doesn't go to the brain. And also it lowers the temperature so that whatever the poison and the impact of the poison, the brain and other major organs is reduced. So I think the anaphylactic response is it's the last desperate action, because the poison, whatever it is in the blood. And now the sole goal is to prevent that poison getting to the brain. So induce as much leakage so the poison gets diluted and gets into the tissue, it doesn't go into the brain, and then they're lowering the temperature. So that's my guess as to physiological role, because it's not a good thing. But I think when you compare lesser of two evils, that's my interpretation. I don't know.
Thank you. So in the same line, do human, lose temperature when they anaphylax as well?
Yeah. So in human, the response is often a sweat response. That's their way. They don't really drop so much. It's mostly sweat. And it's interesting, in our case, when we give capsaicin, which is a TRPV1 activator, it triggers in mice that rapid drop in temperature if you just give them that, because they activate TRPV1. In humans, you take capsaicin, you get the sweat the pepper, and you start to sweat and all. So it's all part of that thermoregulatory response that I think is happening. So there's no big drop unless it's very serious, but it's the other responses. And the body gets cold and clammy because there's sweat, but then the body gets clammy when you're going to anaphylactic shock.
Thank you. Another question from Irakche Suzo Gatstelu. Thank you for this beautiful talk. Would the inflammasome component be also involved in non-IgE dependent degranulation, such as the one controlled by MRGPRX2?
Yes. So the answer, we have started doing some work with not IgE, but other activators, and they are involved in that degranulation. So regardless of whether they are treated with LPS or not, these are involved in, for example, MRGPX2. Yeah, so but not caspase 1. It is ASC and NLRP3. And they both bind CD63 on the granules and move them outside.
Okay. When you have a food allergy that triggers systemic anaphylaxis, it's a little bit different than if you get, a bee sting and then you get and you're allergic to like bee venom, for example. So meaning that when you ingest the allergen, then the first mast cell that might encounter the allergen is actually the one in the gut. So in this system, do you think that also locally in the gut there is this neuron mast cell interaction? And do you think that gut mast cell can also produce IL-1 beta?
I'm going to think yes, based on all the work we've done, but we have not specifically looked, and that's what we are hoping to do. That whole story with the physiological relevance of IL-1 beta production, we have not done any experiments to validate that, so which is where we are going now. So your question is very good. We don't know right now. We're just based on these preliminary data, but we definitely need to look specifically at the mast cells in the gut and look at their capacity to produce IL-1 beta. My suspicion is yes, and there's a lot of neuronal involvement in the leakage, the vascular leakage, the gut epithelial leakage that you see, that's well documented. So, and the mast cells being right there underneath the epithelium suggests to me that they are next to blood vessels and the nerves, and that crosstalk that I'm describing, I think happens, but has not been demonstrated. So it's a very good point you're making. And till that's confirmed, this is just a hypothesis.
Okay. And maybe one last question to conclude this webinar. So if we think about, a disorder in which you have an overproduction of mast cells, like mastocytosis patients, so we know that they are more sensitive to anaphylactic reactions. And sometimes also in the absence of, known allergens, they can go in shock and anaphylaxis. So do you think that all those phenomena that you describe are also true for malignant mast cells? And that would explain actually some symptoms in patients?
Yeah, maybe. Because with these malignant mast cells, there's so much changing in the wiring, their behavior. So I don't know, but it's possible that some of the sensitization and priming that I've described may have a role in aggravating them. But it's, till we do the experiment, we don't know. But it's possible some of these pre-treatments, increasing sensitivity, and all that may play a role.
Okay. And I just see that we have one last question. So more general question about mast cell granules. You said in the introduction that the granules are very long time stable in the tissue. They can stay a long time, and then they will progressively release mediators. So do you think it's a partial release over time or an explosion of granule at one time?
Okay. So we have seen that, so first of all, they're stable. Why do I say that? If we collect these granule remnants, we put it at room temperature in the lab at 70 for 70 days, and you go back and they still maintain that structure. That's in vitro outside. In the body, we see that after degranulation, they're there for a few hours. We don't know, then they disappear. We don't know what happened to them. But what we know is when the mast cell granules are released, the histamine and so on, which are loosely associated, will come out first. And then you get TNF, which is more tightly associated, they are released later. And then like the kinase and so on are much more in amounts, but they tend to be released last. So we see them, and then we don't see like an explosive release, but we see them and then we don't see them. It's not like candy where you get smaller and smaller and smaller. We don't see that. And we think in our studies many years ago, we found that if you activate mast cells, say in the tissue, the granules can traffic through the lymphatics into the lymph node, and you can capture them in the lymph node. But then the lymph node, they immediately disappear. Once they get there, within a few minutes, they disappear. And we think because they have heparinases in the lymph node, they chew up the granules, and that releases like TNF and cytokines and so on. So maybe in the body too, they're there for a while and things are being released, but then something happens and then boom, we don't see them anymore. So something that holds the granules. So remember, the granules don't have a membrane around them. It's all electrostatic forces holding them. And so the contents are slowly released, but then something else happens, and then boom, we don't see them.
Excellent. Thank you. Thank you so much, Soman, for the presentation, also the game of answering questions. And I think everyone is very happy. And I see a lot of applause in the chat. So thank you again. Thank you, everyone, for attending. And I hope you enjoyed it. If you have any further questions, please send them over. And I hope to see you to the next webinar session. Thank you, everyone.
Thank you. Thank you.